So it's a great privilege and an honor for me to welcome our next speaker. She's a biologist, but also a technologist, as you will see. She will give us a glimpse into the pretty cool engineering that goes on at Miltenyi Biotech, both hardware engineering, but also software and wetware engineering. So please welcome with me Dr. Rueß.
Yeah, thanks for the introduction and for having me. I was asked to give you a little bit of context about what the software, how Qt is used, and I'm working for Miltenyi Biotech, and here you can read our mission, but our mission in a nutshell is make cancer history, and this is of course a big mission and a big vision, and we are doing small and sometimes bigger steps towards this direction. So the topic of my talk is proteins, microscopes, and QT, how modern biotech fights cancer. So to also give you a bit of my background, I studied biotechnology. I have a PhD in immunology. I worked for over 10 years in a system integration department, so bringing biology, hardware, software together. And since 2 years, I'm working as a software project manager at Miltenyi. I will give you some insight into the Miltenyi company and provide you an overview of about some applications. And some people here in the room are from QtDev, are working for us, and they might recognize the one or the other slide. So Miltenyi started in a garage, so basically also, and with this in 1989 it was founded, and it is all based, or we took off on an invention by our founder Stefan Miltenyi. And to go back one slide, you see that is a washing machine, and he produced those nanoparticles in that washing machine. So yeah, that is Stefan Miltenyi. He still owns the company, and he studied physics in Cologne, at the University of Cologne. He discovered this method to make iron oxide-based nanoparticles, and then he did a rotation in Dr. Radbruch's lab, which is the immunology lab, and he started then the company in 1989 with the age of 29. And this is the first publication it is all based on, published in Immunological Methods. And the basic principle about this is that you have your cells, the target cells that you are interested in, and they get labeled with an antibody that recognize this, specifically this epitope on the cell, and that antibody is conjugated with this nanoparticle, with an iron oxide nanoparticle. And if you apply the mixture of cells and antibodies with the nanoparticles conjugated on this little column which is packed with beads, iron oxide beads, and you apply a magnet, your cells of interest get stuck and held back in that column, and then you simply remove that column, and then you can wash out your positive cells. So that is I like the earlier talk where you say it is simple, it's technology, but it's so simple the biologists loved it because it was very useful in the lab and provided fast and easy method. So in a nutshell, I already said the company was founded in 1989. We have more than 3,900 employees. We have 50 nationalities. We have 25% of us working in the R&D, like me. We have 46,000 scientific references, 70,000 products, more than 100,000 cell therapy procedures, and 6,000 patients treated with cell products, and we sell in 23 countries, and we serve 73 countries. These are the locations where we are located, so mainly in Germany, also in the US, and one in China, and this is where software development happens. So we are distributed, so software development happens mainly in Germany as well as in the US. So the first instrument I would like to present today is our Maxima instrument. It's basically this instrument. It's a microscope, and some people here in the room might even be colleagues of mine who worked for that software, who developed that software. And this Maxima instrument is, by the technology, overcomes spectral multiplexing limitations. So it is about fluorescence microscopy, and so with this you have, if you think of a microscope, you have this slide and you have the tissue on the slide and a tissue slice, and then you stain that slice with antibodies again, but not this time conjugated to nanoparticles. They are conjugated to fluorochromes, emissioning in the different, with different light, spectral lights. But it is quite limited because you can't just stain your tissue once with those 5 different colors, and the information you get out of this kind of microscopy is quite limited. So we made this Maxima technology, and when you have, first of all, you have a dark field, of course, and then you need to stain with antibodies. You take a picture out of this, so you can stain at once with 3 to 5 antibodies. You make that image, and then you can basically erase those this fluorochrome light, and then you take another stain cycle, do an image, and erase, and so on. And this is a circle, and we have, our record is 400 cycles that last several days, and we have not limited the end, not reached the end. So that is a record currently. It is the limitation are the chemical reagents that we need to provide. And now if you think about 400 cycles, each time maybe 3 to 5 antibodies, this is about 1,000 data points that you get out of this experiment. And I always thought, my goodness, that poor PhD that needs to analyze that data and needs to make a clue out of that. And because of that, we have now this MaxIQ view, which is a nice, very nice software to analyze that data. And now I'm starting off a slideshow with all those different features. So that is an image analysis software, 4th pillar of the Maxima imaging platform family, the last step of the high contact experiment, and this gives the meaning to all this data. So, so this is specifically designed to analyze those hundreds of images, fully integrated into the acquisition instrument. It has an extensive portfolio of analysis tools in one package. It is interactive through image, plots, table, and workflows, and makes it easy to build workflows. So now, just to give you some nice pictures, because this is also very important for microscopy to see actually the tissue, but also then the next is to make meaningful statistics so that you can show significant difference between untreated or treated or cancerous tissue or non-cancerous tissue. So this is a segmentation method, and then we can do gating with this. So this is basically the tissue, but you can, each data point or every single dot over here represents a cell, and then you can make those gatings out of that. And then we can have those distance maps out of this. And yeah, biologists love those kind of pictures where you can really see the different segments. And now, this is the data that the PhD needs to analyze. And we have this very complex workflow editor that you can think of many, many different ways how to analyze your data. So, and more colorful pictures, I like it. Okay, that was one very recent development. Of course, long-lasting projects, we talk about years of development. And, but on the basis of this development, we also have other projects in the pipeline that are bearing to have new software. And one of these is the lab automation system. And this lab automation system, the project was kicked off during the pandemic times in basically spring 2020, and it is a collaboration with and funded by the government of North Rhine-Westphalia. It is shall help for SARS or shall has the intended use to have SARS-CoV-2 T-cell diagnostics. So it's about to detect if your T-cells ever had contact to SARS-CoV-2, if you have specific T-cells that fight corona, basically. And so this is a normal picture. One of you, the one or the other, might have seen it, where you can detect the viral load. And then at the beginning at the disease, and then afterwards you can detect your antibodies, the different kind of antibodies. But then everything, so the level of antibodies decreases and you would not detect that anymore. But the T cells are different. So CD8 is one kind of T cell, and CD4 is another kind of T cell, and they are likely positive. You can detect them for quite a while, and this was actually published already in 2020. The pandemic is very, how to say, it is very dynamic, so must be our development over here. And so we still look for how to integrate that into the in vitro diagnostic that can be offered to the patients. So that is the lab automation system. It has basically, over here you have, these are two liquid handling robots. It has an incubator. Over here is a centrifuge. Then we have a decapper over here. We have also two flow cytometers that detect the fluorochrome labeled cells. And this is how it looks like. That was our first prototype. Still a lot of cables floating around. But we also have this little nice video that I would like to show, where you can see those two decappers, liquid handling systems. Here you just put the sample on, so the blood collecting tubes, and then that is a barcode reader that detects the barcode. The reagents, so the chemical liquids get detected, and here we can see it pipetting. And here it collects the tube. The collection tube goes down to the decapper, and here it gets inverted, shaked. So basically the cells are still alive, and we need to have them alive because they get stimulated with a SARS-CoV-2 protein, and then they show they're alive and have seen that before and get enhanced. That was the incubator. You saw that little door opening. The suspension, that is a shaker, and then arm moves the plate into the centrifuge down where the washing happens. And then it gets transferred to the flow cytometer. So there where the detection happens, the detection of the cells, and now the needle arm comes down and sucks in the sample. Okay, so that was one example. And the next one is actually also, I'm actually very proud of the next sample because I was the product owner of that platform for a year, I think. And it is all about, it's called the Taito. It's a cell sorter. So, and the Taito itself, it is basically this instrument built around this cartridge. And with this cartridge, we sort cells. So what I showed you before with the nanoparticles, now we can also do based on fluorochrome antibodies that are conjugated to fluorochromes, and there we have can reach much more purity and also have more markers that we can select on. So if you look at this cartridge, you can you have this input chamber over here, and then it all goes through this little chip, which is depicted over here, and it is big as this. This is a microchip, and I have some examples over here. And if you come to our booth, you can take a closer look at those very tiny chips. And then how it works is that the cells are going through this channel and then go into the positive. After they detected positive, a little valve opens, I will show you in a minute, and then the positive cells go into that channel, and they land up in this positive collection chamber over here, and those cells that you don't want to have, they get collected in the negative collection chamber. So please, now you see this little video, how the valve opens. So this is really microscopic level. So it is absolutely tiny, and we can sort with this 20,000 cells per second. So this is not like, this is like this. So here you see how the cells get detected by the lasers, and then the green ones get sorted into the other chamber. Again, very simple technology, very simple, but yeah, the technology is quite amazing in my perspective. This is also a little animation how that all goes through, so air goes through this chamber and then presses down the liquid through the chip. It's also sterile. This is always very important when we talk about these applications because this allows us to cultivate the cells and then also re-inject them into the patient. And then this little animation one more time. Okay, that was one very nice example. And now I would also like to come to this, I have to say, our blockbuster at the moment, the Prodigy, because these are our, let's say, bigger steps towards our mission to fight cancer or to make cancer history. And this, it looks like this. It's actually a quite old instrument, I have to say, but this is nice about having a founder that is interested in or believes in technology and believes in the application he has a vision for. So this instrument is the only instrument that can do fully automated manufacturing of patient or of basically cells. So it offers a broad range of application to manufacture patient treatments in a clinical environment. And the intended use is that it is automated sample preparation to produce enriched or depleted cellular products. It transduces and transfects cells. That means basically genetically modifying cells. And then it also offers near to infinity variations. And the initial release was in October 2013. So here comes some background. And what makes this instrument so successful at the moment. So for this, I would like to give you some background in this CAR T-cell therapy. So the indications are currently mainly blood cancer, so leukemia, especially B-cell lymphomas, get targeted with CAR T-cell therapy. And we have some first approaches with solid tumors. And the first step is that the white blood cell collection from the patient in this kind of blood collection tubes. And then that is a starting material for this Clinimax prodigy. So basically, you apply that back on one of those things over here. And what happens then is that the instrument, and this process also takes quite 1 to 2 weeks. So the instrument itself can separate the cells, can genetically modify the T cells and reprogram them in a way that they recognize the cancerous cells. So at the end, the cells get re-injected to the same patient and then can cure the cancer because the CAR T cells recognize the cancerous cells. And we have, I also have a little video how it works. So that is the instrument itself. It doesn't look that fancy, but I have to say it's really, we are the market leader in this company. We are the only ones that provide this from sample to inside solution. And then what I like about that, you see now the centrifuge, so it is also centrifugation, so that is always needed when you separate that. One of the first steps to separate the white blood cells from the red blood cells. And there you can see the back is just gets hung in, and then it gets sucked into the instrument. And then there are many pumps and valves that work over the different days and incubate and apply reagents, apply chemistry, and then incubates for days. And you can also, in between, take a sample, go to your flow cytometer, and analyze the intermediate result of this. And of course, this is a medical product. So this is all it needs to be sterile. Everything needs to be very well documented. It is all happening in a clean room. So the people that use our software are wearing masks, they wear gloves, and absolutely, there should not happen any errors because this is patient material, and the instrument and the software need to work very, very stable. We have a couple of clinical studies ongoing, and they are looking very promising with pharma partners. And this is my favorite slide, I have to say, because it shows the different success stories and gives some pictures behind the numbers. So we have, and this is the most important number over here. We have a 34 to 40% success rate for lasting remission. So that is quite a big number in this field of cancer treatment. And with that slide, that is what was my favorite slide. I'm at the end of my talk. I have no idea about the timing, but thank you for listening. And I don't know if we have time for questions, but we do. Simply.
Are there any questions? Yes, please. Do we have a microphone for the? One comment I wanted to make, pretty much all the software is Qt and C++ across the whole company. Just for the record. Sorry, I think we're grabbing a microphone. I'm sorry, Tobias.
No, it's fine. Wasn't prepared. Thank you.
Thanks for the presentation. I want to ask, I don't know how involved you are in there, but how do you do the testing? Who volunteers their blood to test this thing when you're doing the testing?
So it's not that much blood. So when I worked in the lab and did my immunology PhD, I was there mainly healthy men. My boyfriend volunteered to provide blood, so that was all good. And of course, when the patients, and when you're in a clinical study and the patient has no choice, other choice, they are willing to provide blood for this clinical study, and that might be one of their last chances.
Any other questions? Please. Yeah.
Okay.
And once it gets out, I'm assuming maybe assumption is wrong, but how do you deal with possible bugs that may come out, or do they come out ever? Or if they do, what's the procedure for dealing with those things? Because it's already out, it's being used on patients. Do you see if it affected the patient in some, ill-mannered way, or how does that happen?
How we deal with bugs, that was the question.
Yeah, let's
right?
say there's a bug in the system and somebody said, hey, there's this bug and there's a patient.
So we are in a regulated environment and we have processes for that. We have processes for everything. We have a couple process that deals with bugs, and we have a complaint process how we deal with that. Of course, critical bugs that interrupt and lead to a crash are always very serious for us, and we need to, we then build basically task forces to get those bugs fixed, and sometimes those task forces are holding on for months to get the bugs fixed and get the patient treated. But of course it happens, and this is software, so bugs exist also for those kind of software. Yeah.
More questions?
Yeah, kind of follow-up question. Do you use like a certified version of Qt? I don't know if something exists like that.
Certified version?
There is no certified version of Qt, so Qt being a framework usually does not get certified itself because the product gets certified, and then Qt is one of the components within the product. As far as I'm aware, there is some work going on in Qt company, maybe one of the guys from them can comment, the idea of kind of helping with the certification in a sort of pre-qualification or pre-certification, providing additional documentation materials. There is some thinking in that direction. Nothing exists that I'm aware of, so basically every company does that themselves, but honestly, the way these processes are set up, they probably want to do that themselves also. Yeah. But I mean, Qt as a framework is kind of, you kind of certify the end product, so that's why Qt, there is some things that could be done to make it easier, but you could never like do the whole certification for the customer.
Yeah, I mean, dependency management in certified software is really difficult. So.
Yes, and I mean, that's Qt is just one of many components in such a system. So there's another question there.
Which one?
Thanks. I guess with all the little robots and things, you have also a lot of little microprocessors, and then the user panel for like turning the machine on and off, and at the end you would probably have the scientist who wants to plot the data in his favorite on his laptop or something.
Yes.
So what are those interfaces, or who defines them? Where's the Qt part, and where's maybe the microprocessors involved, and at what stage you would have different other software coming in from,
So who defines the APIs? I have to say that I'm looking into the audience of if my tech lead is available here. Right? So basically, we have so for this big instrument that is 4 meters long with all the different. James Turner I don't know. James, are you here? No. So he rolls out all the interfaces, and he defines that. Of course, it has a lot of business logic in there, and that comes from the business side, so my side, and basically, who defines the APIs?
I mean,
to phrase it differently, like I guess there's some microprocessors that don't have Qt, and then at some point maybe there's a embedded Linux or whatever that has the Qt, and at some point there's Windows PC or whatever for the scientist. Like, do you have software to control the instrumentation things also on your favorite Windows PC, or is it all on the Linux PC, or do you sometimes have it on the microprocessor level? Or?
It's both. So it's both embedded and desktop. And there are use cases where you want to do it on the device, so controlling the actual microscope that happens on the device, but also parameterization might happen on the desktop. There's data manipulation going on the device, but also later in diagnosis. So you have that actually referring back to what Dario was saying, it's a digital product in the sense that you interact with it all these products you interact with in a variety of ways during the experiment, before the experiment, after the experiment, on the device, off the device, in the cloud. So the data flows through various systems. It's not like, it is one machine in a sense, but the overall user experience is very much more complicated than that and distributed across.
I mean, the challenge then always, I guess, is that you don't develop all the software, but there's like different parties coming in, and then you have to rely on them working for you, or like adjusting the software for you, or.
And it's even worse because we provide our software to our customers, and they have their own self-made laboratory information system, and we have a standard HL7 that they should follow, but this is so open that. Yeah, it is possible that we need to have a customized version for those customers, and if they pay us sufficiently, then of course we will do that.
It's even worse than that, because people write their own Python code to pre-process the data, to post-process the data. They write Python code to automate parts of the system themselves, to integrate into other systems, to export and import. So much of the software actually also provides Python automation so that the users can program this into their own workflow. So it's a programmable system in that sense. So it has whatever software complexity you could think of, it's in there. But yeah, the interface is obviously a big problem in all of this.
Thank you for your presentation. And I work with medical devices too, so I know that you have to do lots of tests and provide proof that your software never does something wrong. So I was wondering, are you able to do a lot of automated testing, or do you also have to rely to a lot of manual integration testing and stuff like that, that I know we have to go through that as well. So.
Yeah, so very good question. We aim for automated testing, but we also always have a part of manual testing, especially because sometimes you need to put something onto the instrument and see it if it's moving and doesn't crash. And so it's a mixture of automated and manual testing, what we aim for.
My question would be more from biological or engineering part. The cell separator that you presented, what is the error rate allowed for this sorting of the cells, and how do you calibrate it?
How do we calibrate this? So the lasers get calibration, so they have the intensity value that we have fluorochrome labeled beads that we can detect if the lasers are working. The error rate, I have to say, so you say the question is about how often does the instrument fail, or what?
More like it's not that it is 100% correct,
okay.
right?
Okay, so that depends a bit on the application and also basically also on how big the cells are and how well they fit through this channel. And you can also choose between different applications. Do you rather want to have high speed or time doesn't matter, then purity matters, then we can reach a purity of 99% cells of one type, so T cells, for example, that can be reached.
Thank you.
In the back.
So you have shown 4 products from one company, and each product looks completely different to the other. So are the teams have same code classes, some same design classes, and do they have their own solution for the same UX, user experience problem? Or does they have their own solutions always?
Thank you for that question, and you see me smiling because we are establishing, so at the moment many of those products have their own little life, and we're also bored, and then they bring their own software with them. But we are establishing a software platform, and they shall have the same code base and shall have the same software, shall share the same platform modules, and by natural, they get the same UX and UI design. We are about to establish that, and the first instrument that I showed you, the Maxima, will serve as a blueprint for that. We are not there yet, but you might imagine that, if with a company that size and with very different groups, as you said, it's about a lot of communication, bringing teams together. Everybody needs to get the understanding, and terminology is the key over here, I have to say, so that they all have the same understanding. What is an experiment? What is a procedure? What is a command? And many meetings are just about getting the terminology right here. And then we can think about, yes, we share the same code and have the same UX design.
Are there any other questions? If not, then thank you very much, Anna. That was very, very enlightening. So thanks for coming.
Thank you.
Thanks,
and
everyone.
here are the chips.